the past and a little bit about the future. It will be, I think, somewhat technical for the second part, just because I think the one important outcome will be to, as you've already heard, some of the people are thinking about therapeutics and comparing methodologies, and at least being aware of approaches will be worthwhile. But also, I was last here 2 years ago. Couldn't make it last year, although of course the Sieber Center was here. And I looked at my slides, and it's pretty exciting about some of the advances. So the first look back, look forward. This slide has now been shown by several people today, but here's a quiz for the families. When you do a proximity labeling of HP1, what genes come down? Just kidding. But you've heard that I think Alex mentioned about you can pull down this protein over here, and what comes down as part of a complex is CHAMP1, POGZ. I'm going to talk about ADMP. I think when we met 2 years ago and we talked about bringing in the POGZ families, we also talked about ADMP, and I know Jeff spoke to the ADMP Family Foundation, right? So I think it is really amazing that there is a joint conference right now. We can talk, it's a subtle thing, it's not exactly the same pathway, and we've already heard some of the discussion about that, but it's still better to be together, right? Other genes that are also pulled down, like EHMT1, it's a so-called Kleefstra syndrome gene, also in the same complex, CHD4, there's the developmental disorder that also is a resultant of this. So quite a few genes in this complex are associated with NDDs, neurodevelopmental disorders. All right. Irini talked about the Autism Sequencing Consortium. The reason we know about all of these genes, or one of the reasons, is this consortium that was created more than a decade ago by me and some of my colleagues, and we systematically sequenced thousands of individuals with developmental disorders. This is the first paper in 2014, and you could see POGZ and ADMP are somewhere in there. There's ADMP, there's POGZ. CHAMP1 didn't come up so much in the early studies, partly because CHAMP1 does have a slightly different phenotype than what we were collecting. But this is how we know these genes are actually directly involved in developmental delay syndromes. Over the years, the Autism Sequencing Consortium, as well as the Simons Foundation, have continued to sequence. I won't bore you with the details, but this is the 2020 paper that Irini showed. She showed you this little panel here, and this is the most recent one, and we have another one coming out this year. And if you go back to that slide of these various genes, you can see that that is ADMP, that is POGZ, that is CTC4, which is also in here. And this is a study in England focusing more on early developmental delays, and there you see CHAMP1, EHMT1 as well. So, there is a fairly broad, we think there's over 1,000 genes that when mutated will produce a developmental delay syndrome. You've already heard that the phenotypes overlap, but they're also not specific. They're not really like, oh, this is the same thing so much as this is what it looks like when we have a problem with development. All right, so I think just that you have put together CHAMP1 and White Sutton together, I think is a really big deal, and I applaud you for doing that. Oh. Do we have different colors? Is it blue for one side and yellow for the other? Oh, I didn't know that till now. Looks like everybody on the CHAMP1 side has gone home. We only have, look around. It's only the White Sutton. Oh, not this table. Okay. So last time in 2022, we presented some high throughput methods. So if I have 1,000 genes, how do I understand them biologically? Do I do it one by one, or can I do dozens at a time? And this study we presented in 2022 just came out. It's now on the same bioRxiv server that you heard about before. And I won't belabor it, but looking at 60 genes at a time, well, I can't see it, so you certainly can't see it. But POGZ and whereas CHAMP1 and EHMT1 all have the similar property of making nerve cells differentiate more rapidly in this assay. Again, I would be very careful about saying it's a similar phenotype, but in terms of nerve cell development, brain cell development, in this specific assay on a high throughput way, 3 of those genes in that little complex are having the same effect of pushing brain development quickly. Sounds like a good thing, but it's not, because you need everything to develop at the right time. You can't have nerves waiting for something and already be developed before the signal comes. So I just want to close the chapter on that. One of the reasons it took so long is the methods that we have, the analytical methods we have to analyze these complex systems, actually, they're not any good yet. And so we spent a lot of time developing a new analytical framework to study brain development from a molecular level. See what I said about CHAMP1 going home? They're already rolling up the... Okay. So with all of these genes, right, there is an opportunity, and this is where we're going to pivot to the future, to think about them as rare genetic disorders or orphan diseases. And that is something that academia does actually very well, and historically probably better than biotech and pharma, because biotech and pharma are afraid to take the leap, right? I won't go through this, but there are kind of standard methods of using, well, you've already heard, if you have a gene, and so we do this discovery for genes, if you have a gene, you can make cells in culture that carry the mutation. You heard a lot about that. You can make mice or other animals that carry the mutation. You heard about that too. And of course, you can bring in families with the mutations, and you can study the phenotypes, and you can develop biomarkers, and you can hopefully begin to think about therapeutics. You can test in your cells in culture, test in your animal models, and think about deploying. And so the way we do that at the Sieber Center is we actually have, we have about 60 people, maybe 16 faculty, and about 60 or 70 people working across the labs that take a parallel approach. For a gene we choose to study, like CHAMP1, we have ongoing clinical studies at all levels, and we have ongoing studies with the same genes in cells and culture and animal models, with the idea of understanding biology, drug repositioning, or finding drugs that already exist, testing them in the preclinical model, and then ultimately trying them clinically, and developing new drugs. And you've heard about this whole thing in the context of the day. We try and do it in the same institution, side by side, at the same time. It has a certain power to it. The most important thing, this is from a white paper that was put together at the NIH. We all know that treatments in the brain are hard. You've heard about intrathecal injections and LMP and getting things into the brain. One of the critical things that the FDA asks is, how are you going to assess improvements, right? So we talk a lot about natural history studies, for example. And last time we were here, we presented, the work that Tess did. Tess, somewhere over there. Where's Tess? Okay, Tess is gonna, why don't you stand up? There you go. She'll never talk to me again. So, this was a protocol that was in the height of COVID. We developed a protocol to do a remote assessment, and it was applied to CHAMP1. We've subsequently, in these rooms here, in room 31 and 32, we validated some of the remote assessments by some of the CHAMP1 families coming back. And so we began to develop a clinical battery to understand the phenotypes and develop endpoints. One of the things that happened last time I was here, when there were lots of people from the Sieber Center here doing assessments, it was to answer this question that has come up today multiple times, right? Which is, does CHAMP1 act through haploinsufficiency? And I think the answer, sorry folks, is definitely no for 99% of the mutations, and probably 100%. And so, and the reason we know that is because there is in fact a haploinsufficiency syndrome that Koza talked about, the 13q34 deletion syndrome. And if you just look at some standard scores, these are things like IQ, where 100 is the mean, okay? If you have a deletion across the entire gene, this standard score, developmental quotient, IQ equivalent, is in the 70s and 80s. If you have a point mutation in the gene, it's in the 40s and 50s. That is a huge difference. It cannot be ignored, right? So, Tess did this work. Where's Tess? Why don't you stand up? It's critically important because it tells us about the genetic mechanism, and you can't develop therapeutics for genetic disorder without understanding the mechanism. Obviously, Marcella and her team also made similar observations. I think we kind of are pretty clear about this now, and I think also the advisory board has made that clear as well. Okay. The other challenge we've heard about is delivery to the CNS. How do you get something into the brain? And we have a recent, this is why there's so much optimism. Spinal muscular atrophy is a disorder that in many cases the child will not survive after the first couple of years. A new therapy, one of these oligonucleotide therapies that you just heard about from Chris, was tried and approved in 2017. Those kids are still alive and doing well. Within a year, another company developed a gene therapy that mostly replaced Spinraza. And the gene therapy is kind of what you've also heard about from Hawkins Laboratory, about an AAV that replaces the gene. And then a year after that, somebody came up with a small molecule that works like the antisense oligonucleotide to cause a change to do a gene-targeted therapy. So if you were in the business world and you spend a few $100 million making this, you're kind of out of business here, and then you're kind of out of business there. The truth is that they're all being used, all three approaches, but that over the course of a couple of years, three radically different approaches could come to the clinic for this devastating disorder and change lives, change the whole field. The other thing that changed the field is, of course, the COVID LNPs, the way we get the vaccines into our bodies. Everybody's talking about, can you package the ASO, as you heard from some of the questions before, in these same things that go to your COVID vaccines? And the short answer is yes, and the other answer is now the FDA realizes that these kinds of methodologies are quite safe. So we are trying to develop these kinds of approaches for the various disorders, and that's me. So we're very, very happy that the MUSC group is willing to collaborate on this. We have some ASOs, and here these ASOs are specifically designed to be what's called allele specific. They are meant to, you want to get a picture of me in front of the picture? Allele specific means they're meant to disrupt or to knock down the bad version, if you will, and leave the healthy version intact, right? And that's a strategy that would actually make sense here. So we have some proof of concept going on there, and I won't talk about that. The other thing is, it turns out that you can take small pieces of DNA or nucleic acids, and you can trick the machinery of the cell and say, when you see this mutation, don't put the mutation in, put in the right amino acid. Okay, it's called suppressor tRNA. And basically what it does what I said, it sounds like magic, right? There's a mutation in the DNA, it's supposed to be making a protein, and you put in a tiny piece of tRNA, and instead of it making a mutation, it makes the regular protein. It's actually on the RNA level. So I had the slide for CHAMP1, and then I added the one for POG-Z. This is, if you go to ClinVar, which is one of the repositories of clinical genetic information, you can see that of the pathogenic or likely pathogenic mutations in the gene CHAMP1, and you look at the smaller ones, not the deletions, the vast majority, and Kozo said this too, are nonsense, and the rest are frameshift. There really aren't any missense variants that a hard-boiled clinical geneticist would currently say is definitely causal, right? So nonsense mutations can be fixed by this method of suppressor tRNA. And if you look at CHAMP1, there are multiple, these are the ones that happen more than once. A lot of them are recurrent. And so going back to Stefano's lab, he has some of these arginine mutations and so on that we could say, let's see if we can fix them, right? POGZ, we have even more mutations in POGZ, and there are plenty of nonsense mutations, and some of them are similar to the ones in CHAMP1. So the idea then is, can you take these suppressor tRNAs and trick the cell, instead of putting a stop codon, to put in the right amino acid? And we're trying this arginine to stop. So this, these individuals have an arginine, the R, like a pirate, or here's an arginine too, and that amino acid is turned into the asterisk, which is a stop codon. So that's a mutation. So what you want is not to put the star there, but put the R back, okay? Isn't there some like pirates world around here? So we want to turn this stop codon that shouldn't have been there back into an arginine, or this tyrosine back into a tyrosine. And just to show you proof of concept with, all right, so one of the ADMP gene is part of the family. Welcome to ADMP gene family. There is a very common mutation in that gene that makes a tyrosine into a stop. We made the suppressor tRNAs for that tyrosine, and if the mutation is there, you don't see any cells lighting up. But when you put in these suppressor tRNAs, each little ball here is a cell expressing a fluorescent protein. The fluorescent protein contains the mutation. It contains a tyrosine that turned into a stop, but we're tricking the cell to ignore that and make the proper protein, which is fluorescent. It's kind of, I'm sure that didn't make a lot of sense to many people, but it shows you that we can take this little tiny piece of nucleic acid and tell the cell, instead of putting in a mutation there, correct it. Okay? And now we have to do it with real proteins. The other thing we've done also with ADMP is you've heard about stem cells and organoids. And you can use the phenotypes, you can use the changes you see in the organoids to screen drugs. So you've heard, for example, about the electrophysiological changes from the CHAMP1 mutations, right? So that could be used as a screen for drugs. So this is ADMP again. These are just organoids that are stained and viewed from on top. And you can see that at this level of development in the organoids, there should be a nice green patch in the middle. The green patch is called PAX6. And instead, if you're missing one copy of ADMP, there's less PAX6 staining. And you could actually screen 10,000 compounds, not easily, but this is how much of that green inner circle is there in the wild type. This is how much is in the mutant, carrying one mutant copy of ADMP. And this is the results with 10,000 different drugs, one on each well. And what you see is, you should be here, the mutation brings you here, the mutation plus drug, some of the drugs bring you to where you need to be. And we now have 33 drugs that, some of which are actually overlapping in mechanism, so we're optimistic about that. This is something that's fair, we are now very skilled at doing. The only problem is it's enormously expensive. The other thing we've been working on a lot is targeting RNA, just like you heard before, but now with small molecules. And this is actually, I almost left this out because it's really more, because it's not a haploinsufficiency for the most part, but it can be used for POGZ. Basically, you've heard about the tails of RNA, and it's called the UTRs, right? And Chris was telling you about using antisense oligonucleotides to target that. Well, it turns out that small molecules actually bind the same structures. And you can make reporter systems where you have something that gives off light, and then you can throw drugs on, and did you make the same amount of product, more or less? And that turns out to be a pretty easy screen relative to the other one I showed you. And here's the results for ADMP again, but we've already done it with 3 different proteins. We found a bunch of compounds, small molecules. Small molecules is like all traditional medicine that you take as a small molecule. We know how to get it into the body. We know how to get it from the body into the brain, right? So this is like a big deal in terms of getting the drug into the brain. Here are 2 compounds from the first screen. We call them hit compound 1, and then we call the second one hit compound. And this little fuzzy bar here is ADMP, the gene we're interested in. And as you increase from here to here, the amount of these compounds, the amount of protein in the cell goes up directly, okay? So we're directly seeing, so ADMP, just like with POGZ, is a haploinsufficiency. You're missing half of the amount, right? And so you want to push up, if you can, the levels. And so far, we did a pilot in 4 different genes, and for all 4 of them, we have compounds that can drive up the expression of the endogenous protein. So, early days, but another approach that we think is going to be useful for this family of disorders. 5 minutes. Can I finish now, or do you want me to keep on? I was going to end and just say thank you, but Jeff just told me I should talk for 5 more minutes. Oh! All right. I'm going to stop anyways.
So technically, time was up when you were starting, right? Because
just a comment, and I saw Matt Disney up there. So Scripps are now partnering with Florida. They have our iPSC lines. He couldn't come today, but you're piloting that program with ADMP, and what was the other gene that was up there?
You're working with?
For the small molecules with Dr. Disney. Are you collaborating with him on that?
Yeah. So the 4 in our pilot were ADMP, SHANK3, gosh, I can't even remember, FOXP1 and DDX3X. And we have hits for all of them, and we have a grant in to do more, including CHAMP1.
Okay, just letting you know he's starting to do the work over there too. We can talk about that later.
All right, but okay, it's probably good for us to talk before, because if he's thinking it's a haploinsufficiency, we need to correct that, right?
Great talk. I'm really interested in the haploinsufficiency part, like the IQ score difference is really striking, but I'm wondering, do you have any cellular expression levels? Because I'm gonna try to think out loud. Could it be that a full deletion, the cell perceives it as having half of the amount of protein, so it really drives up the expression, while in a mutation, the cell in absolute levels of the protein does not see anything different, so there's no stress to produce more functional protein, but half of the proteins are non-functional. That's why it may be like more severe. Do you have any expression level differences?
People have looked in blood with mutations, and it really does seem to be on the RNA level of haploinsufficiency. These dosage-sensitive genes tend not to have that kind of feedback, at least universally. I mean, the reality is that we're never 100% sure of anything, but I think we're at least 90% sure. And yes, there are possible mechanisms that we could still be very wrong, but we always have to go with our best data, especially when you're thinking about drug discovery, right? I try and make the point that all of us scientists love the tail. We love the most extreme thing that like blows everything else out of the water, but when you're thinking about drug discovery, you want to go with your best knowledge, and 90% is, that's all you're ever going to get, right?
Hi, it was really a great talk. I was really curious about your drug screen on the Firefly luciferase reporter. Mm-hmm. That's you're transfecting organoids with those constructs, or could you elaborate a little bit? And also, given what we know that like the endogenous chromatin in the brain is like not opening enhancers up as it should at the right time and place, do you think the drugs? Is there any way you could think of another way to screen drugs that takes into account the endogenous chromatin?
Yeah, I mean, listen, I mean, luciferase screening is, it's cheap, it's quick, and it's not biological at all, right? So we immediately pivot after luciferase screening to try and see, but the luciferase screening here is asking a simple question, right? It's saying, when I target the UTR, am I getting more of the RNA, okay? And then once you have that, then you can immediately pivot and say, okay, let me take those same, and there's a whole bunch of controls you do, but then you turn around and you say, once I have that, and this is in HEC293 cells, so again, it's not ideal. Then you can just put the same small molecule in cells in culture with endogenous ADMP and show that the protein is upregulated. Should we be doing it in iPSCs? Yes. Are we? Yes. Do I have the results? No. Should we be doing it in mice? Yes. Well, the problem with the mice UTR is actually not the same, so we really can't do it in mice. So the screen is just to get to 20 molecules to try further things on. The nice thing about small molecules is that if it works at all, we know so much about medchem to make better and better molecules that can get to right places in the body. Yeah, and one of the things you can do very easily, of course, we already have structure-activity relationships. We already know which moieties on these molecules are driving efficacy. So it's just a screen. And the second question was, yeah, kind of the same thing. We're not touching any kind of DNA structure, right? We're just saying, can we get more? And then, if we can, when do we have to get it in to make a difference?
Right, so that the suppressor tRNA that you showed us that converts a nonsense mutation to an arginine, that sounds dangerous. I mean, isn't that gonna convert other regular stop codons into an argine? And you're gonna
So far it doesn't, and we don't really know why, and that's what's dangerous, that we don't know why.
Yeah,
so
well,
if you do,
yeah.
nowadays, if you throw in one of these suppressor tRNAs, you see no read-through or very little read-through
of normal proteins.
Of normal proteins.
I see.
Now,
Weird.
I want to know why, because some of the genes we're talking about, like CHAMP1 or ADMP, the mutations are in the terminal exon for the most part, right? And if it's somehow because it's recognized in the terminal exon, it may never work in certain genes. Mm-hmm. So here's a challenge to the audience. Please figure out why that doesn't happen. If we throw in these, the question is, we have these mutations that we can correct, but there are every other protein in the body has the same kind of termination codon that we don't want to correct. And yet somehow when we throw it on cells or in vivo, you do not see read-through.
Well, some normal protein translations, there's 2 stop codons at the end, so that would be a stop, stop gate too.
But generally, so far generally, people don't see read-through. Yeah. It's weird, and we really need to understand it, especially when we're talking about genes where the mutations are, for the most part, in the terminal exon. Oh, there'll be one more talk after me.
So actually going back to what Dr. Gianni was saying with the nonsense mutation, for us, I have a missense mutation, but with our variant, the glycine got switched with valine. So I don't know if y'all have seen that, where the GLY gets mixed in with the VAL. And what are y'all doing in the future research to try to correct that?
We're going to crack everything. That's the good news. I am of the opinion that we need one win, and then we'll finally get the industry involved, right? So, everybody wants to know that you're working on their specific mutation, but imagine if we succeeded with any mutation, right? And somebody got a lot better, then it would change the field completely, because then the industry, biotech and pharma, would say, oh, we can do that better than you can, Joe. Please get out of our way. And they would go ahead, right? So we just need, we need to have a win. And it almost, that's why we're trying methodologies that are even mutation specific, as you point out. And that's okay, because if it works, somebody else will figure out the next mutation. That's my faith. That's my belief. I have pretty much given up hope that pharma is going to jump in without that first bit of evidence. But with that first evidence, they'll tell all of us, you guys don't know what you're doing, get out of our way. And we'll retire. And that'll be nice.
Anybody have any last questions?
Thank you.
Thank you so much.